Hello everyone, my name is Matthias Körschens and I'm from the Computer Vision Group of the Friedrich University, Jena, Germany. My talk today is about weekly supervised segmentation pretraining for plant cover prediction. First, let me give a quick motivation why we are doing this. So, plant community compositions are a very important indicator for environmental changes and are therefore a very important part in plant biodiversity research. While we nowadays have the means to record plant communities over longer time spans completely automatically, for example using video or image data, we still do not have the means to also automatically analyze this data, and a manual analysis is not only extremely laborious but also subjective. Therefore, having an automated system to do this work for us would enable objective, high quality and high temporal resolution data. So the task we are actually trying to solve is the so-called plant cover prediction task. And yeah, the basic aim is to utilize images like the one you can see on the left side, analyze the plant species contained in the images, and then predict a plant cover vector, which is a numerical vector like the one you can see on the right side, which tells us how many % of the ground are covered by each different plant species. You can see that this kind of analysis is quite complex, as the plant species can look very similar. They have different sizes, and of course, as you can see in this image, they also strongly occlude each other. While analyzing the occluded parts of the plants also plays a huge role in the correct prediction of the plant cover percentages, in this talk we will focus on the analysis of the visible parts only. The reason for this is that for predicting the occluded image parts correctly, we first need a good analysis of the visible parts, which we can then use to reason about the occluded areas. So for the basic approach we utilize here is the one we already introduced in a previous paper, and this approach basically comprises of a neural network with two parts. The first part is a network backbone or feature extractor, which gets as input a plant cover image and outputs pixel-wise feature descriptors. These feature descriptors are then used by the network head, which then calculates the pixel-wise probabilities of each plant species, which are then aggregated into the total cover percentages, and the network head also generates a segmentation map for each image, where the user can also check the correctness of the results. As mentioned before, images like the one you can see on the right side are very complex, and therefore training only on these does not lead to optimal results. Therefore, we utilize pre-training and transfer learning with isolated images, which are usually freely available on the web. So pages like, for example, the one of the Global Biodiversity Information Facility usually provide images of isolated plants like the ones you can see on the left side, and also provide the respective class information, in this case plant species information, which we can utilize for training. However, training the network or pre-training the network using only classification data has multiple caveats, as we found. For example, standard classification networks, like for example a ResNet50, usually have a very small output resolution due to their network structure. And this makes a correct localization of especially the very small plants extremely hard. We can counter this by using a feature pyramid network in conjunction with the ResNet50. However, there's also another caveat caused by global average pooling during classification training. So training using global average pooling usually generates rather fuzzy feature maps due to the averaging over the whole image. However, there's also another caveat here, which is the extreme focus on the most discriminative regions. In the image on the left side, you can see, for example, the class activations of a network trained on classification data. You can see here that the network primarily focuses on the flowers or blossoms of the plant and less on the leaves of the plant. However, as you have seen in the plant cover images before, we mainly have images containing primarily only leaves of the plants and not flowers at all. Therefore, if the network focuses on the flowers of the plants, this does not have any advantage for us. And therefore we propose a solution to this in the following. So the basic idea of our approach is to somehow convert the class information we have into segmentation information, so that we can basically mark the complete plant, including the leaves and flowers, completely in one image, and then simply train a segmentation network to reproduce this so that the network focuses on all the regions more equally than during classification pre-training. And the weights of this training can then later on be used in our plant cover segmentation network so that the results are improved in this way. The base of our approach are so-called class activation maps. For those who are not aware of this approach was introduced by Zhu et al. In 2016, and the basic idea behind this is to utilize a network which was trained with global average pooling and a fully connected layer. And afterwards, the global average pooling layer is removed and the fully connected layer and its weights are repurposed into a 1x1 convolutional layer, so that the network simply displays at each location where which class was located, which can then look, for example, like below. With this method, we can also convert the classifications into segmentations in the following way. We first have a trained classification network and let it generate a class activation map as the first step. Of course, for all the images in the training set, we also have the class information. Therefore, we can simply remove the irrelevant classes and purge it from the class activation maps, which leaves us only with the class activation maps of the class in question. And we can then simply binarize it by thresholding into foreground and background, so that we have simply the segmentation of the plant, like the image you can see on the right side. So this is our complete pipeline. It comprises 3 steps or 3 phases. The first phase is standard classification pre-training, where we utilize the classification laws and simply train the network on the plant species information. This network then outputs the weak segmentations, which are then used in the second phase for segmentation pre-training, where we utilize these segmentations for training with the segmentation laws. And the weights of this network are then simply used in the third phase, where we apply the plant cover training and utilize a regression loss on the plant cover percentages. And this network is then able to output the respective cover percentages as well as the aforementioned segmentations. In our experiments, we utilize 2 different datasets. The first one you can see on the left side. This dataset is our pre-training dataset, and it contains images from the Global Biodiversity Information Facility. We constructed this dataset ourselves, and it contains 750 training images per plant species, and it contains images of 8 different plant species. The dataset you can see on the right side is our actual target dataset, which is called the InsectArmageddon dataset, named after the eponymous IDIF project, which took place in 2018. So this dataset was already analyzed and several analyses published in the last year. And of this dataset, we utilized 682 images of 9 different plant species. To actually be able to analyze the quality of the segmentations our network generates, and therefore also quality of the analysis of the visible parts, we had to annotate a small number of images from the InsectArmageddon dataset by hand, 2 examples of which you can see here. As you can see, the plants are usually quite small, and of course they are also very hard to distinguish sometimes from one another. And it's not only a hard task for the network, but also for the humans as well. We can then use these segmentations simply with an intersection over union metric to evaluate our approach. and we do exactly this here. Here we see the differences in the intersection over union when switching from classification pretraining to segmentation pretraining. On the left side, you can see the absolute changes in intersection over union, and you can see there's a general positive trend for almost all of the plants, except for grasses, for example. And we can also take a look at the relative changes for the plants on the right side. And there we can see that, for example, Achillea millefolium and Lotus cuniculatus gain an intersection over union of more than 100%, which means the recognition rate of these plants basically doubles. We can see one large drop here for Scotonoides autumnalis. However, this is explained quite easily. This plant is quite rare in the InsectArmageddon dataset, and it was almost not contained in our segmentation data, and therefore there was almost nothing to detect, and this drop is basically only a drop from 0.01 to completely zero, so this drop is negligible in this case. Taking a look at some qualitative results, here we can see on the left side the segmentations generated by our network using classification pretraining, where we can see that the most dominant plants in the dominant plant species in the dataset, for example Trifolium pratense here in green, and Plantagula lanceolata in turquoise down here, are segmented correctly in most parts. Most parts of the segmentations are still quite all over the place. However, if we take a look at the segmentations generated after segmentation pretraining, we can see that, for example, on the left side in pink, Achillea millefolium is also additionally segmented correctly here, as well as Centaurea yatsea in purple in multiple occasions, as well as the plant species mentioned before in the other image, and grasses on the right side in blue. So overall, we can see a drastic improvement in segmentation quality using our approach. So to summarize my talk, we introduced a novel pre-training pipeline which generates segmentations on the fly and utilizes them for segmentation pre-training, which in turn outperforms the standard classification pre-training. Also, we have just seen that the segmentation of the visible parts improves when utilizing this approach. In the future, we would like to apply this approach in alternative areas. So, for example, it might be applicable in general segmentation tasks, as well as in general fine-grained classification tasks. And we would like to extend our approach to also include the occluded plant parts, as of course, as I mentioned before, the occluded parts are very important in the analysis as well. And with this, I would like to thank you for your attention. I would like to thank my two working groups for their support and the Carl Zeiss Stiftung for their financial support. Thank you very much.